Dear students, this is our 24th lecture on bioorganic chemistry, and today we will discuss about FAD and FMN as coenzymes under coenzyme chemistry. The flavin mononucleotide and flavin adenine dinucleotide. So these are the two very important coenzymes which are derived from vitamin riboflavin, and they are also known as flavins. So the enzymes which uses flavin as coenzyme, they are called as flavoproteins. So flavoproteins, they are basically enzyme coenzyme complex of flavins. Then the vitamin riboflavin is a component of two closely related coenzymes, that is FMN and FAD, which catalyze many types of biosynthetic reactions, and this belongs to a group of yellow fluorescent pigments which are called as flavins, as I have already told you. The flavin ring is attached to an alcohol related to ribose, which is ribitol, and this was first isolated from milk. And the orange yellow color of flavins is due to this isoalloxazine ring which is present in these coenzymes. Their heat stability is relatively higher, but they are very much destroyed by light, so they are to be protected from light if they are to be made catalytic in nature. These FAD and FMN, they catalyze many types of oxidation reduction reactions in presence of enzymes. So these are the chemical structures of these FMN and FAD. Flavin mononucleotide is having, this part is flavin, and this part is the alcoholic part, that is ribitol, and this part is the phosphate which is present in the coenzyme. And the main activity of this coenzyme is due to this heteroatoms, nitrogen heteroatoms present at these positions. Similarly, this is a part of riboflavin vitamin B2. So if we attach phosphate group to vitamin B2 riboflavin, then this is converted into FMN. Similarly, flavin adenine dinucleotide is that if the vitamin B2 is attached to two phosphate groups, in addition we are having adenine attached to these two phosphate groups, so we will get flavin adenine dinucleotide. But the point of activity in both the coenzymes is same, this ring containing nitrogen as the heteroatoms. The FAD and FMN, they are very much used for oxidation of succinate to fumarate. That means the saturated carbon-carbon bond is converted into unsaturated carbon-carbon double bond. So we can say that alkanes, they are converted into alkenes. So typical example is conversion of succinate into fumarate in presence of enzyme succinate dehydrogenase, and this FAD or FMN, they act as coenzymes for this conversion. So basically what happens is that these are the active centers where the reduction will take place. So this will be reduced to FADH2 or FMNH2, depending upon whether we use FAD or FMN. Whereas here in case of substrate, the oxidation will take place at this place. So one hydrogen from here, one hydrogen from here, they will be shifted to one hydrogen to this nitrogen and one hydrogen to this nitrogen, and this carbon carbon single bond will be converted into carbon carbon double bond. So this is how the oxidation of the substrate will take place in presence of these two, any of these two as coenzymes. Then biochemical function of FAD is that as you have seen that most of the flavins, they participate in redox reactions. Redox reaction means oxidation reduction reactions, and these flavins, they are critical for metabolism of carbohydrates, fats and proteins. And as I have told you, the riboflavin functions as tightly bound prosthetic group of a class of dehydrogenases known as flavoproteins or flavin dehydrogenases. And in case of flavins, this ring is isoalloxazine ring is the active part where the conversion of the substrate will lead to either formation of oxidation products or reduction products by using a pair of hydrogen atoms removed from the substrate molecule. So succinate dehydrogenase, as we have seen in the previous slide, is an example of flavin dehydrogenase. And the general biochemical function of riboflavin is that it helps in the formation of antibodies and red blood cells. It maintains cell respiration, and this is also required for the maintenance of good vision, skin, alleviates eye fatigue, and promotes general health in case of humans. So these are very important functions as far as human body is concerned. The flavoproteins, they catalyze a wide variety of biochemical reactions. For example, if we use glucose oxidase as enzyme and FAD or FMN is used as coenzymes, then they can undergo dehydrogenation reactions. So alcohols, they will be converted to carbonyl compounds. Then amine oxidase enzyme will be responsible for deamination of various substrates using FAD or FMN as the coenzyme. So amino group will be oxidized to carbonyl group. Then succinate dehydrogenase, we have seen the example. The carbon carbon single bond will be converted into carbon carbon double bond. So saturation will be converted into unsaturation. So dehydrogenation will take place. Similarly, NADH dehydrogenase enzyme in combination with FAD will be used up for dehydrogenation. So here NADH will be converted into NAD plus. So reduced form of NAD will be converted into oxidized form of NAD. Then glutathione reductase, this also is used for dehydrogenation and cyclization of dithiols to these types of cyclic structures they are formed. Similarly, if we use para-hydroxybenzoate hydroxylase, this is used for oxygenation of phenols. So you can see over here, this phenol is hydroxylated at the ortho position in combination with FAD as coenzyme. Then the radical reactions which take place in case of FAD, they can be either 2 lactone reactions or 1 lactone reactions. And semiquinones, they are formed as intermediate, they are stable radicals with unpaired electrons, and these unpaired electrons, they delocalize through the isoalloxazine ring, which is the main activity point in the flavins. So for example, if FAD is reacted with a free radical, then this will add to nitrogen 5 of this isoalloxazine ring. So we will get this type of radical anion which will absorb at lambda max 480 nanometers. Then this radical is a stable radical, and this will interact with H positive ion, and we will get semi-quinone radical anion which will absorb lambda max 570 nanometers. So this will appear blue, this will appear red. Then this anion will be converted into this type of anion, where negative charge on oxygen is transferred to nitrogen with the reaction with another electron. So this is one electron and this is two electron reaction, and through this two electron reaction, FAD will be converted into FADH2. So this will be reduced to FADH2. So basically, if you see, we have added two hydrogen atoms, one at this nitrogen, other at this nitrogen. So this is reduced form of FAD. Then mechanism of action in case of FAD is that most of the flavins, they bind non-covalently with the enzyme, and this binding is very strong, which can be seen from the equilibrium constant, which is of the order of 10 to the power -8 molar. And various types of reduction and oxidation reactions take place like this, that enzyme flavin is converted into protein and flavin, and this enzyme flavin FAD reacts with the substrate, and the substrate is oxidized to X, and these two hydrogens, they are transferred to the FAD, and this is how reduction of FAD will take place to give us FADH2. Similarly, oxidation of FADH2 can also take place when this will react with a substrate Y. These two hydrogens, they will be transferred to Y, and Y will be reduced and FADH2 will be oxidized. So this is how different types of redox reactions, they can take place with flavins. And when electron acceptor is Y in the oxidation, the half reaction is commonly molecular oxygen. So this molecular oxygen will react with one electron, and we will get a free radical of this type. Then with another electron, this will be converted into hydrogen peroxide if two electron reduction is taking place. Similarly, if 4 electron reduction is taking place, then after these 2 electrons, 2 more electrons, they are added to H2O2, then this will be converted into water. So this will depend that what type of product is formed, whether H2O2 is formed or water is formed, depending upon that how many electrons they are used, either 2 electrons or 4 electrons in the completion of the reaction. Then various reactions of FAD. FAD can undergo alkaline degradation. Riboflavin on reaction with alkali is converted into this type of carboxylic acid with the removal of urea from here. So urea is removed with the breaking of this ring, and we will get 4 substituted 6,7 dimethyl 3-oxo 3,4 dihydroquinoxaline 2 carboxylic acid and urea as the product when riboflavin is hydrolyzed at room temperature. Similarly, riboflavin reacts with sulfite, and the reaction takes place at nitrogen 5. Nitrogen 5 sulfite is formed on reaction with riboflavin. Then photoreduction of flavins can lead to the formation of different types of products. Either leukodeuteroflavin is formed or deuteroflavin is formed after one lactone or two lactone reactions, depending upon the reaction conditions. In the first instance, when UV light is irradiated on flavin, the singlet state will be converted into a excited triplet state of the flavin, and then a diradical will be formed like this. One H from here will be eliminated as H radical. This will form a radical at the carbon, and then one radical will be formed at nitrogen 5 with the movement of the other electrons to give us this type of diradical. And this type of diradical can either undergo coupling reaction. So this electron, one electron from here, one electron from here, they will form a carbon nitrogen bond at this place, and movement of this will lead to the formation of a radical at this place. So this radical and this radical, they can combine to form a carbon nitrogen double bond, and this is how this radical will be converted into leukodeuteroflavin, having this type of cyclic structure. And this leukodeuteroflavin can be oxidized at this position to give us these types of ketones as the product with the breaking of this carbon nitrogen bond, and we will get the deuteroflavins as the final product. And these deuteroflavins, they can also be formed directly from this diradical by disproportionation method. So this will undergo this H radical is removed from here, then H radical is removed from here to form a carbon oxygen double bond, and this will form a carbon nitrogen double bond, and this will move to this place. So this is how this diradical can be converted into deuteroflavin as the product. Then photodealkylation can also take place. The flavins, they can undergo these types of cis periplanar conformation intermediates to give us these types of duochromes as the product with the elimination of a ketone from this place. So dealkylation results in the formation of this lumichrome, and a ketone is formed from these types of reactions. Then photoaddition can also take place. Photoaddition takes place in this way that when flavins in their ground state, they are oxidized with alcohols under UV light, we will get these types of hydroxy or alkoxy dihydroflavins as the product. So these types of reactions, they are called as photoaddition reactions. Then photoreduction and dealkylation that generally occur through triplet state, whereas photoaddition takes place through excited singlet state, as you have seen over here. So this is how photoaddition will lead to the formation of alkoxyl flavins as the product. Then photosensitized isomerization can also take place, that when flavins in the ground state, they are irradiated with UV light, they will first be converted into excited singlet state, and then this excited singlet state will be converted into excited triplet state. And in the excited triplet state, these flavins, they will interact with the substrate, and they will reach again to the ground state, and this substrate will become excited in the triplet state, and in the excited state this will further lead to the formation of cis trans isomerization products. So this is how we can get different types of cis trans isomerization reactions. Then similarly, the flavins, they can undergo photosensitized oxidation. Again, flavins in the ground state, they are excited to their triplet states through UV light, and then when substrate is reacted with these excited triplet state flavins, they come back to their ground state, whereas substrates, they are converted into products. So for example, in this alpha amino acids, they are oxidized to carbon dioxide and aldehydes and ammonia as the product when flavins, they are reacted with UV light. Similarly, oxidation of dihydrolipoate to lipoic acid can take place when we use FAD. FAD will be reduced to FADH2, and cyclization at this place will take place through dehydrogenation in the presence of enzyme dihydrolipoate dehydrogenase. So this group is not affected, only dehydrogenation will take place at these sulfur atoms. So mechanism of this reaction is that one of the sulfur atoms will act as a nucleophile and attack this carbon of the isoxazoline ring, and this double bond will abstract this proton from the active site of the enzyme, and we will have an enzyme coenzyme substrate complex which is formed in a covalent manner. So a covalent coenzyme substrate complex is formed. Then in the next step, this will act as a base, active site of the enzyme will act as a base. This will abstract this proton, and cyclization will take place to give us lipoate as the product, and FAD will be converted into FADH2. So this is how oxidation of dihydrolipoate takes place. Similarly, oxidation of amino acids also can take place in presence of enzyme D-amino acid oxidase or L-amino acid oxidase. So FAD similarly will be reduced to FADH2, and amino acids, they will be converted into amino acids. So mechanism of this reaction is this base from the active site of the enzyme will abstract this proton of the amino acid, alpha amino acid, and this will be converted into a carbanion. This carbanion will attack the coenzyme, and a coenzyme substrate complex which is covalent in nature will be formed, and then this nitrogen will attack with its lone pair to generate this carbon nitrogen double bond, and this bond will be broken. FAD will be reduced to FADOH2, and alpha amino acid will be oxidized to amino acid. So this is how oxidation of amino acids to amino acids can take place. Thank you very much.